Hi, hello, welcome back. So today we're going to start a new unit. It's evolution, and even though we think it's a new unit, there's nothing new on this. Basically, we're going to incorporate a lot of the elements and a lot of the concepts that we have been studying all year long, and starting to see the big picture. So up to now, we have been focusing on molecular aspects of how living organisms function, and now we are going to start integrating all those molecular components and see how all those little reactions that are happening inside our bodies contribute to the successful life, if you wish, of an organism. So evolution is the first. Today what I'm going to try to do is show you the different pieces of evidence that we have for the process that we call evolution. And by definition or by common sense, evolution basically means change over time. If you ever play Pokemon, Pokemons evolve, natural process. The more they fight, they learn new things, and they have to evolve. Well, it's the same with all living things. We are all evolving all the time. We are all incorporating new things and changing. It's very rare that anything remains the same constantly all the time. So that's what I want you to get, that evolution is nothing else but change of organisms over time in response to their environment, pretty much. That's the premise. So I have a nice link to a video. It's about 8 minutes long. I really would like for you to see it. It's very entertaining and very, very thought-provoking. I encourage you to watch it maybe at the end of this presentation, and later, maybe after we have studied evolution a few more days, because you're going to see a lot of elements that you're going to start understanding even better. So it's painful mating and the evolution of felines. So if you're a cat person, you have to watch it. Definitely. All right, so here we go, evolution by natural selection. And just remember that the person that proposed this process is basically Charles Darwin. And Darwin got all the ideas from basically a trip that he took when he was only in his early 20s. He was offered the opportunity to travel around the world in the Beagle, that's the name of the boat. Took several years and basically started in England, covered all South America around, went around, stopped right there, those are the Galapagos Islands, went across the Pacific, kept on going, touched Australia, South Africa, went back to South America because they needed the proper winds to go back to England, and from there they returned to England several years ago. And during the whole time, Darwin was kind of the naturalist of the trip, and he was constantly collecting and observing and taking notes on everything and reading a lot of the literature that had been published up to that day, not only in the sciences but also in the economics world and business. So a lot of time to read and a lot of time to think. Now, Darwin, as you know, a lot of his ideas just kind of all of a sudden came when he was in the Galapagos Islands because of what he saw there. But at the same time, I want to point out another place in the world, that's the Malaysia, basically, because at the same time that Darwin was doing this, not at the same time, but around the same time frame, another scientist, Alfred Wallace, was working here. He was collecting specimens, mostly birds, for the British Museum. And in this group of islands, he started noticing exactly the same patterns that Darwin had noticed in his trip around the world. And basically, he came up with the same idea about how organisms change over time and what's driving this change. And even though they both came up with the same idea kind of at the same time and was published technically at the same time, Darwin is the one that gets most of the credit because he's the one that had the most evidence about this process. Darwin's tome is a huge book, one piece of evidence after another. Wallace, as I said, was working mostly with birds, and so had limited, but again, same concepts, same principles, same ideas, but Darwin gets most of the credit because he's the one that developed the idea the furthest. So just a little bit of history to understand the process. And if you summarize Darwin's or Wallace's principles, basically they propose that natural selection, natural meaning the environment, is the one that determines which species are going to survive, and depending on which ones survive, those are the genes that are going to be passed to the next generation. If you are dead and you don't reproduce, your genes are gone. So the premises basically are that for natural selection are that basically there is variation in the population. Organisms are different, they look different, they have different traits. Basically also, organisms over-reproduce. We have more individuals that can survive because resources are limited. Because resources are limited, this is going to lead to competition among organisms for food, for mating, for nesting sites. You have to escape predators, so it's going to be a rough world out there. And because of this competition, there's going to be differential survival. Not everybody gets to survive, and only a select group is the one that survives. And this group that survives is the group that has the traits that allow them to survive under those environmental conditions. These traits that allow you to survive under specific environmental conditions at a given time, right now, this is what we call adaptations. Basically, adaptation, anything that gives you an edge and that allows you to survive, that increases your chances of survival. Now, if you increase your chances of survival, you are going to reproduce, and your good genes, your good adaptations get to be passed on to the next generation. That's the premise of this. Now, if your genes, the good genes, get passed to the next generation is going to look different from the previous one, because a lot of those that couldn't reproduce are gone, and those not so well adapted genes are gone too. So every generation there is a little bit of change because only a select group gets to pass their genes on, because they are the only ones that get to reproduce. So that's the last premise, the differential reproduction concept that allows only a select group to reproduce, and that is going to produce this change over time because not everybody gets to pass their genes on. This is the essence, and I would like for you to apply this to everything that you do in your life, because you're going to see this process everywhere. So, let's go with the evidence that is supporting evolution, all the data that we have, that we keep on collecting, and how we are studying this process. There are actually 5 pieces of evidence that I'm going to show you. If you want to make an outline, number 1 is the fossil record, that is going to show us 3 things basically: that species change over time, that species disappear, they go extinct, and that there are what we call transitional forms, basically those things in between that give us a clue that this is a constant process. Then we have the anatomical record that is going to include homologous and vestigial structures that basically give us an idea of common ancestry. Embryology and development, again, give us an idea of common ancestry, how organisms are related. Then we have all the evidence from the molecular record. Now that we are able to sequence proteins, sequence DNA, we can compare the DNA of the different living organisms, and based on that we can infer and we can figure out their evolutionary relationships. Obviously, if I'm more closely related to somebody, our sequences are going to be similar. If I'm more distantly related, they are going to be more different, because through time, mutations accumulate and allow our DNA and proteins to become more and more different. And the last piece of evidence that I'm going to try to show you, it refers to basically clear examples of natural selection that we are seeing in real time, and also what we have done, artificial selection that humans have been doing for at least 10,000 years since the development of agriculture and domestication of animals. So examples, so we are going to start now one by one, try to go over all these examples. So number one, fossil record. So what do we have in the fossil record? Please remember that the fossils are basically are usually found, not usually, pretty much found in what's called sedimentary rock. Sedimentary rocks are the ones that are formed by the accumulation of sediments, one layer on top of another. And when organisms die, if you die and you are quickly, and that's the key, quickly covered by a layer of sediment. So I have my cell phone just died, and layers of sediment start accumulating on top of this. Basically, that organism over time is going to be covered, it's going to be protected, and the organic components are going to be replaced by the mineral structure of the sediments, and you're going to become a fossil. So requirement number one, it has to be buried very quickly, and this usually happens in oceans, estuaries, lakes, places where you have a lot of sediment and usually water, or deserts where you have, not necessarily deserts, but places where you have things like sandstorms or anything that can cover you very quickly. If you don't get covered, what happens to you? I die, I sit there, and organisms eat me up, and you disappear. So not everything gets to fossilize, and so that's why the fossil record is kind of a lucky chance that the organism died and was fairly quickly covered by sediments. So keep that in mind. Not everything fossilized, and if you have anything that is hard in your body, that definitely increases the chances of you being preserved. So since fossils, organisms die, they are covered by sediment. Basically, these layers of sediment accumulate, and that gives you an idea of the age of the organism. If you are closer to the top, you are younger. Basically, you lived more recently. Deeper down, basically, that shows farther back in time. Now, if you look at the fossil record, you're going to see that organisms have changed, that organisms like these beautiful trilobites existed for a very short period of time, of about 30 or 40 million years, and then they totally disappeared, never again, totally extinct. Same thing with things like ammonites. Fossils, please don't only think that these organisms sometimes are the leftovers of the organisms, like these beautiful coprolites that are basically feces, fecal material, that fossilize. And these are extremely useful because they allow us to figure out what kind of things these creatures were eating, and the size, because based on the size of the coprolites and the shape and the structure gives you a great insight into the physiology of the organisms. Of course, animals are not the only ones. Leaves fossilize beautifully. So all these are clues that we are going to use that tell us, yeah, there were creatures before that we don't have now, and even the creatures that we have now, they have been changing over time. So have an example here for elephants, and this is a new chart that you're going to start becoming familiar. Notice that usually here is going to have an indication of time. So we have 0, that's the present, and goes back down to 34 million years. And over here, it shows you kind of the relationship between the different organisms. Here we have the two elephants that we have, the Asian and the African elephant, right here. And if you notice, right there is when they have a common ancestor. And that happened about 5 million years ago. So this is telling us that about 5 million years ago, there was one type of elephant, and that in the last 5 million years, these two species have become more and more different. Now, we all know mammoth, very familiar to the mammoth. If you look at the mammoth and you see how they fit in the whole scheme, the closest relatives of the mammoth happen to be the current elephant. Then if you keep on going with the fossils that we have found, notice that these other, like Stegodon, existed about maybe 10 million years ago. And if you notice the structure, kind of similar but not quite the same, going back to the very first elephants that were much, much smaller, that lived around 34 million years ago. More evidence from the fossil record that tell us how organisms change, and the horse is a prime example. If you look at the graph, this graph here is showing us body size. Please remember, data is very important, and you have to be able to understand how this graph, what the information is showing you. Body size in kilograms, if you want to figure out pounds, roughly multiply by 2. Body size in kilograms, and millions of years ago. So 0, that means now the present. The one only horse element that we have now here is equus, that is the horse, donkeys, and very closely related. The horse itself has changed significantly. Notice the first relatives of the horse. The body size was much smaller, but through the fossil record we can see that there is a tendency to increase in body size. So we can get this kind of evidence from the fossil record. Change in body size, but that was not the only thing. If you look over here, the most primitive fossils, or oldest fossils that we have of the body, the horses were like the size of small dogs, kind of, German Shepherd kind of size, small size, and they have four toes, and this is the pattern of the teeth. What happened through time? You can follow the evolution of the foot in all these fossils, and if you look at the structure of the foot, you're going to see that there is a tendency towards a reduction of 3 of those toes, until you have, in the modern day horse, only 1 toe. The 3 other ones, and you can see how they're getting smaller through the fossil record. Same thing with the teeth. The teeth have increased with more rough surfaces to increase the chewing capacity of to be able to break all the grass that they eat and chew it more efficiently. So all this you can deduct from the fossil record and figure out how the horse has changed and what they looked in the previous time. Also, the shape of the teeth again gives you more indication about the selective forces and the type of diet that they have, and probably an indication also of the plants available at a given time. So species change, species get extinct. Now, one of the major things that fossils are helping us now, as we are able to dig in different places and get lucky, because to find a fossil you have to dig, and where you dig sometimes is pure luck. And some of the fossils that we have unearthed fairly recently include, for example, the Archaeopteryx that is now considered what they used to call the missing link between birds and reptiles. You know that birds evolved from reptiles, but was like a very rough transition there. You have a reptile, a bird. Well, here we have somebody that kind of looks in between. These species that help us to see all the little steps, we call them transitional species, because they show the transition between one group and another. So this is Archaeopteryx, one of the first reptiles that had feathers. The role of the feathers early on was not necessarily to fly, but more for insulation and protection. And we know now that this is the first step towards the evolution of birds. Other transitional species that we have include a Tiktaalik that we are going to see later on. That is a fish, basically, where the fins, you can see the bones actually that lead from the transition from fish to amphibians, where we can start having upper body limbs. So transitional species is very important because allow us to see the transition between organisms. The only problem is hard to find because it's pure luck. You have to dig, 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 and you never know what you're gonna get. So we don't have them because not everything gets fossilized, and because they are all underground, and the only type of rock that you can find them is sedimentary rocks. So first you need to know your geology to know where to dig. Anatomical record. Here we have very interesting sets of structures. We have something that we call homologous structures, analogous structures, and vestigial structures that you need to understand very well. So let's start with the first one, homologous structures. Homo, remember, means the same. Homo, the same, meaning the same ancestor. Same ancestor. So homologous structures show you similarities in the structure of the bodies because those organisms share a common ancestor. So best example, this is the textbook example, is the homologous structure between a human arm, a cat, whale, and a bat, basically any of the vertebrates. If you look at any of the vertebrates, you're gonna see that we use our upper body limbs for different functions, to run, to work, to fly, to swim. But if you look inside at the bone structure of all these different limbs that are used for different things, they all have exactly the same bones. The only difference between the bones is the relative size of the bones, but they all have exactly the same bones, and in this diagram those bones are color-coded. So what in the wrist bones here, in a human we have our wrist bones very small here. Here you have them in a cat, here you have them in the flipper of a dolphin, and there they are in a bat. Now the fingers, the same thing. In us, 5, very useful. In the cat, much more thicker. In the dolphin, notice how they have elongated to be able to provide support for the flipper. Same thing in the bat. Notice that they have elongated, and the membrane, basically very little apoptosis here. The structure remain together, so now it can be used as a wing. Same here, very little apoptosis between the digits. So these are all homologous structures because homo means the same ancestor. We all have exactly the same structure. Now, the difference is that we use them for different things, but that is because we are adapting to different environments. But the fact that we all have the same means that we share a common ancestor. This is evidence of very close evolutionary relationships. So we are all very, very, very closely related. If you want to figure out the names and you are good at this, you can compare all of them. We have sheep, seal, bat, dolphin, shrew. Look at them, they are fascinating. You have all these pictures, very nice and big, in your textbook. Now animals, of course, we tend to favor animals, but you can see exactly the same process of homologous structures. Same origin, maybe different function now, also in plants. Leaves, we are used to seeing the beautiful leaves that do mostly photosynthesis, flat and broad. Remember, we also have needles. We also have the succulent leaves that they still do photosynthesis, but they also adapt to store water. So same origin, but they start having different functions. Spines, again, these are leaves actually, but they have basically adapted to defend the organism and to reduce transpiration, as we will see pretty soon. Tendrils, these are again modified leaves, same origin, so all these plants basically share the common ancestor. But in this case, the leaves are modified into little tendrils that allow the plants to grab onto surfaces as they climb further and up, trying to get closer to the sun to maximize sunlight. And of course, poinsettias. A lot of people think, oh, how beautiful this flower. These are not the flower, these are just colored leaves. The flowers are actually those little buds that you have in there. So, homologous structure, they look different, they are all basically the same origin, common ancestor. Ooh, check this out. You have a nice bird and a dragonfly. They both fly. And you should say, oh, they both have wings. They should be related. Are they? No. This is a problem that you are going to encounter and that you have to be critical and analytical and think about it. Yes, they all fly because they have adapted to a new environment, the air. They have developed these adaptations that give them an edge and can fly. However, do they have a common ancestor? If you look at the structure of a wing here and a wing here, do you see the same structures inside? Can you follow the development? The answer is, of course, no. So these structures that serve the same function but have very different origins are called analogous structures. So think of this, analogous, you start in different points and you come together because the environment determined that you would go in that direction because that's the best adaptation. These are analogous structures and they do not show relationships. They do not, you cannot use analogous structures to figure out relationships between organisms because they have followed different evolutionary paths, they do not have a common ancestor, and you got to the same spot just because of environmental pressures. This is called convergent evolution. You start in different places, totally unrelated, but you look the same. But you look the same because of the environment, because of the adaptations. That shows you how wonderful the environment is, but you cannot use that as an evidence of relatedness between species. Similar functions and external form, great adaptations, but the structures are internal are different and different development pathways. No evolutionary relationship. They only show that the environment is a great force for evolution. Okay? They solve a similar problem starting in different places. Convergent evolution. Think of convergent lines. They start in different places and they come together. So, very good. Keep your eyes on this. Don't be fooled by the looks. Looks are deceiving. So, convergent evolution. Here we have it. And you see that flight, for example, this beautiful adaptation of being able to fly and conquer the airspace for escaping or for moving faster, evolved in 3 different groups. Even though we know that these at the end are kind of share a common ancestor, they evolved, they came to the same adaptation quite independently. So clearly between the dragonfly and these are analogous structures. Dragonfly and bat, analogous structures, very clearly. Okay, so please keep that in mind. They do not have a common ancestor. These two do, but compared to this one, they do not have a common ancestor. So again, analogous structures show you the power of the environment and is what we call convergent evolution, but they do not show common ancestry. And another good example of this is the ability to swim, conquer that aquatic environment. Dolphins at the top of the vertebrate evolution, fishes at the very bottom of vertebrate evolution. And what do they have in common? Fins to move, very streamlined bodies that allows you to move quickly, very smooth skin. Dolphins lost their hair. These guys, of course, have the scales and mucus. So streamlined fins, anything that allows you to move quickly through the water. So these structures, the fins, the streamlined body, is just basically convergent evolution that shows you that particular environments require particular sets of characteristics. last piece that we are still in the anatomical record. One more piece here. It has to do with vestigial organs. Vestigial organs are structures that we have in our bodies that at the present time seem to have absolutely no function. We have them, but we don't kind of use them for anything. So they are kind of leftovers from the past, and that means that in the past they had a function, and whoever else has that structure, that means we had a common ancestor there. So examples of this that we have is, for example, the tailbone in humans. We have a tailbone that we don't use for anything. It's actually just a nuisance for some people, and that's a leftover of our beautiful tail. We have we have an appendix that we don't use very much now, not like other mammals, that the appendix is extremely large and well developed. Ours is very small, and again, a source of problems sometimes. There is evidence that it's used still by your immune system and as a nursery for a growing bacteria for your large intestine, but again, is a structure that is not necessarily useful right now, as proved by the fact that you remove it and your life goes on. Another examples, you have fishes that live in caves. Closer relatives live outside and have eyes. In caves, no light, no need for eyes. It's not like, okay, I don't need the eye, I'm gonna get rid of my eye. If you are in a cave and you're not using your eyes, there is no selective pressure to keep functioning eyes being passed on to the next generation. So, like in everything, you start accumulating mutations. In a normal environment, if this kind of fish in an above the surface river loses the eyesight, this fish is dead and cannot pass on its genes. In a cave, this fish loses the eyesight, no big deal because it's not needed. So it gets to pass on those genes that have been accumulating mutations. So because there is no selection, basically just keeps on going, and mutations that are always happening start accumulating until you get to the point that a structure becomes non-functional, because there is no selective pressure to keep it going in one direction, like the closer relative above ground or outside the cave. So we have the appendix and tailbone in humans. We have the eyes in the blind fish that have been lost. If you look at snakes, just streamlined body. If you look at a boa and you look at the internal structure, you're gonna see that where the hips used to be, where the legs are supposed to be, there are the hip bones still there. You don't see them protruding to the outside, but the hip bones are still there. Why? Because who was the common ancestor of a snake? A regular reptile with 4 limbs. Whales, same thing. Whales, even though they don't have back legs, this is a transitional species, you can see these bones actually highly reduced in whales. Actually, they are not seen outside, they don't use them for swimming, but if you remove the blubber and you look at the skeleton, they still have leftover of these bones. Now, if you look at the fossil record before modern day whales, you look at the ancestors of the whales, like this one, well, you still have those, but notice that they used to have still the little back flippers with the bones from the hip structure, with the femur and everything else. Femur and the 2 other bones in your legs there. So everything is there, but they're not used, but the leftovers are still there. What does it tell you if the bones are still there? It tells you that the ancestor of this had those bones and that the bones have been reduced. So it shows relatedness and ancestry. So they were not always sea creatures, obviously. Actually, cows and hippopotamus are the closest relatives of whales. So finally, the other piece of evidence from the anatomical record, just from looking at the anatomy, is if comes from comparative embryology. If you look at the embryos, the early developmental stages of organisms, you're going to see that there is some patterns that are the same. And if they have the same patterns of development early on, that tells you that they had to be closely related at some point. So I want to show you, in vertebrates at least, there are a couple of structures that all vertebrates have, and all vertebrate embryos are going to have a tail right there. And we all have a structure here that we call gill pouches that are basically kind of primitive gills like the first vertebrates that were fishes. So all of us have those structures, and of course we all have the backbone. That's what it makes us truly vertebrates. So we have the gill pouches there and the tail. Those are two very common structures that we have with all other vertebrates. So if you look at this chart, you can see here we have fish, salamander, tortoise, chicken, pig, cow, rabbit, and humans. First stage of development, if you look at them, we all look the same. You wouldn't be able to tell the difference between one and another. And you can see the gill pouches, clearly that structure that we have all in common, gill pouches and tails across the board. What happens as development continues? You can see how each species is kind of choosing its path, kind of like with cells. You start broad and then you start focusing on your path. As development proceeds, you can see that morphology is changing until you get to the later stages of development, where clearly we start differentiating into what we are. From human, look, we are still not very different looking from other creatures. We are very similar. So please notice that turtles and chickens, very similar again. And fishes with salamanders, again, very similar, which is showing you the relatedness. So we can use all these clues. Please notice that not a single factor can be used to figure out how species are related or that things have changed over time. But all these are clues that this just doesn't happen by chance. By the way, that looks like Franklin. Did you ever read Franklin? That's Franklin. Alright, so we have fossil evidence that shows us change, extinction, transitional species. We had morphology that gives us a clue about homologous structures, vestigial structures, and embryology, that all those show common ancestry. But we also have those analogous structures that don't show common ancestry but show us what environment can do and brings us to convergent evolution. The third piece of evidence that has been developed in the last 20 years and is becoming the powerhouse of evolution right now is the molecular record. And the molecular record is based in the comparison of two things. One is the raw DNA or RNA sequences that we have, and the other one is comparison of protein sequences, those amino acids that make our proteins. To do this, you usually pick to compare molecules that are extremely necessary and that have not changed too much. For example, the two common ones that you need to really understand very well is, one is hemoglobin, because that's a common feature of all vertebrates and other organisms too, the molecule that carries oxygen in our bodies, very necessary. And the other molecule, the other protein that is very important is cytochrome C. Remember that cytochrome C is found in the mitochondria, part of the electron transport chain that in the process of producing ATP. since we all do, pretty much everybody uses this step, we all have cytochrome C from a lot of lowly bacteria. To us, we all have the same or very similar protein in our mitochondria. So we can compare the sequence of this and see how it has changed over time. Now, when we compare sequences, protein or DNA sequences, if you have two organisms and their sequences are very similar, that tells you that you are more closely related. If you are closely related, that also means that the point at which these two species separated to become the two different species that we have now happened fairly recently. So depending on how different the sequence or how similar they are going to give you an indication of how far in time the branches that gave rise to these two different species basically split up. If you are very closely related, very similar sequence. If you are very unrelated because you split up from that branch a long time ago, you're going to have more differences in your sequences. Again, let's look at this graph quickly. Here you have nucleotide substitutions, basically how many nucleotides are different. So here you have 100 nucleotides different. Here you have 0 different. They are the same, they are very different. And here you have this is for hemoglobin, between humans and kangaroos. If you compare the sequence of a hemoglobin in human and kangaroos, what do you find? There are about 100 differences in the amino acid sequence. Now, this difference, not amino acid, nucleotides, sorry. There are about 100 nucleotides different between the hemoglobin for human and kangaroos. Now, what happens? Humans and kangaroos, we are all vertebrates, we are all mammals, but our paths diverged about 125 million years ago. So for 125, we have had nothing in common, and during those 125 million years ago, this is based in the fossil record, we have accumulated about 100 differences. If you look down at the opposite extreme, you go to a horse and a donkey. They have very few differences in their nucleotide sequences, and that is because horse and a donkey, we know that they are very closely related, and according to the fossil record, they actually start becoming different species fairly recently. So less time for change to occur. If you want to stick with humans, human and rodents. We are much more closer to a rodent than a kangaroo. Kangaroos are marsupials, we split long ago. Rodents, we're still kind of on the rodent side. Notice here we have, instead of about 100, we have about 50 or 60. And that is because our lineages starting to diverge only about 75 million years ago. So less time for mutations to accumulate. Please be good at analyzing this kind of data. So here you have hemoglobin. This is number of amino acids different between human and each one of these creatures. So between human and macaques, primates, we have 8 differences. Between human and dogs, there are 32 differences. With the birds, 45, with frogs, 67, and with lampreys, one of the most primitive kind of fish-like creatures, we have 125 differences. If you plot on the bottom the time at which we branched off, when these lineages diverge, you can clearly see that with the macaque, it was fairly recent, just a couple million years, only 8. With the dog, we split. Here is the point where we split, about 15. If you go to the lamprey, we have 125 differences. When did we split from the lampreys? Here we go, up to here we were kind of the same. Here is with the lineages split. we have been separated from lampreys from 80 million years, so plenty of time to accumulate mutations that cause these differences. So this is what we use this data to construct these beautiful diagrams that we are going to start calling cladograms, and that are going to show us relationships between organisms. Right? Now, evidence of evolution, remember I'm showing you that all these things show us how that really organisms are changing over time. From the fossil record to the anatomical record to the molecular record, we see that things are changing. I want to show you this last piece of evidence at the molecular level about primates. And let's look at the number of chromosomes that they have, and you're going to see something fascinating. I love this. Orangutans have 48 chromosomes, gorillas have 48 chromosomes, chimpanzees have 48 chromosomes. Orangutans, gorillas, chimpanzees, right here, 48 chromosomes. Homo sapiens, we have 46 chromosomes. And we know now that these are our closest relatives, that evolutionarily we separated from them only a couple million years ago. So, what happened to our chromosomes? If you look at the nucleotide sequences of the chromosomes and you look at the chromosome structure, remember chromosomes have centromeres and they have telomeres. Remember telomeres at the very end that protect the chromosomes. Remember that they get shorter and shorter and shorter with every cell division. We can test this hypothesis. We can say, ooh, 46, 48. Either we lost it, or what else could happen? You can fuse two chromosomes. Either totally disappears somehow, but if you disappear a chromosome, you lose a lot of information. And we know that our DNA and chimpanzees or any other primate is up there in the 98, 99% the same. So clearly it didn't disappear. So you can test the hypothesis. For example, could these two chromosomes that were different fused and become one chromosome? You know, two strands of DNA, could they join together to make just one strand? Nice theory, and that was a theory that got proposed a long time ago about why we have this difference. Oh yeah, probably we fused our chromosomes. Can we test it? Can we verify for that? With the tools that we have now, yes. And want to see this. These are the chromosomes in chimpanzees and other primates. Telomere, telomere, blue are telomeres, red is the centromere. Blue is telomeres, red is a centromere. If we propose that probably two chromosomes in a primate fused to make one chromosome in humans, this is what you should see. The two chromosomes, they fuse to make a human, and the fuse part here, you should see the sequences of telomeres right there. Remember, telomeres don't code for anything, they're highly repetitive sequences. And you can see telomeres at the end. And also, besides finding telomere sequences in the middle, you also should find two different centromere sequences that are also relatively specific. So, if this hypothesis is correct, we should be able to find and look at our chromosomes and find one chromosome that has this funny pattern of telomere sequences stuck in the middle and somehow centromere sequences that are kind of repetitive. So guess what they found? That exactly that is what happened. Chromosome number 2. Chromosome number 2, the second one in humans, is really the product of the fusion of two chromosomes, two smaller chromosomes in primates. You can go to the original article paper that was published. It was published in Nature, the equivalent of Science, but in England. And basically, this is what the human chromosome number 2 looks like. You have telomere sequences in the middle. No other chromosome has that. And you have two sequences that are similar to what you should have as centromeres. Only one centromere remain functional. Beautiful. Evolution demonstrated by your molecular record. Simple. So that's how contribute to have 46 chromosomes instead of 48. 2 of the primate chromosomes got fused, and that's how we ended up with 46. So totally testable hypothesis that you can use all the molecular evidence for. Finally, I want to present some evidence from natural and artificial selection that actually natural selection and the whole set of principles that Darwin proposed are still working on today. So most of this is a lot of common sense that you have been exposed to, and I need to make sure that you understand the proper processes, because there is a lot of misunderstanding about this. Natural selection in action, we have speed up selection of some organisms right now, and we have selected for resistance to antibiotics, also selected for resistance to insecticides, because we have been trying to kill a lot of these creatures. So here you have insecticide. You know that we spray fields for crop protection. If you have a bunch of creatures there, particular, again, these photographs with colors are very good in your textbook. Basically, when you apply pesticide, you're going to kill pretty much most of them, because pesticides are designed to interfere with some metabolic pathways specific for the creature that you are trying to kill. Now, they work pretty well, 99.9%. Because we know that there is variation within the population. And since there is variation, there is always going to be a few individuals that are slightly different, because they already have particular mutations. We all have mutations happening all the time. So, if you have a couple of individuals with slightly different shapes of proteins or enzymes, slightly different, there is a significant chance that these creatures are not going to die when you apply the insecticide. So the ones that are different, that had those mutations already, get to survive. Everybody else dies. So what happens now with the ones that survive? The very few, not too many, the few that survive. Well, now you have a whole field of crops. You eliminated most of the competition, and now the few of you can survive, no competition, and you can reproduce like crazy. So what happens in the next few generations? Most of the individuals that are going to be living in that area that you just spray with insecticide are impervious to the insecticide, are not going to be killed because they are resistant. We selected a group, and that group, we have no idea what other genes were lost here. The only genes that here we are selecting for resistance, but you have to think, what genes are going with the resistance? All these other genes that we kill here, they are eliminated from the gene pool. So we are changing how organisms, the makeup, the genetic makeup of organisms, because of this artificial selection that we are doing right now, because we are spraying, we are trying to kill, but we are not killing everybody. The few that don't die is because they have the genes. And basically, if it's in the genes, it's something that they can transmit on to the offspring. This is an extremely important concept, that when we spray, we kill everybody except the ones that already have the mutations and the ability to survive. So one generation after another, whatever we're using becomes less and less effective, and that's why we need to come up with new insecticides, new antibiotics constantly, and that's why we have the problem with MRSA, that is basically, this is methicillin, that's one of the strongest antibiotics, resistant Staphylococcus aureus. That's one of the bacteria that is resistant pretty much to everything. So we can do that, and we have been documenting how species are changing with the use of these selective killings. Now, in the natural environment, we were able to see natural selection at work in England, and this is the classic textbook example of the pepper moth. This is a moth that grew in England in the 1800s, well, still out there. But this moth has two patterns of coloration, a dark and a lighter color. In the early 1800s, just go and put everything up. In the early 1800s, this moth in England, if you look at the trees around England, this is before the Industrial Revolution, a lot of birches, and if you know anything about these trees, they are usually light color bark, kind of whitish, and there are a lot of lichens that are also light color greenish growing on these trees. So nice light color trees all over. Now, if you have moths here, which moth has the advantage here? Obviously the light color pattern, because they can hide, basically camouflage in this, and they don't get eaten by the birds. So keep that in mind. Then what happened? Late in the 1800s, we have the Industrial Revolution, fueled by coal. Coal, this is before the time of filters in the factory, so all the soot and black smoke came up, and basically trees and buildings became covered by soot. So the nice, beautiful, light colored bark trees now became very dark in less than 100 years. Everything from trees to buildings. So go nice light, everything dark, very grim to England. Then in the 1900s, mid-1900s, the whole environmental movement, and we start cleaning Clean Air Acts, not only here but in England. We start cleaning the air, reducing environmental pollution. So what happens now? Since we are cleaning the air, new trees are light color again. So everything goes back to light color. So light, dark, light in about 150 years. Well, if you look at the percentage, people have been doing this, looking at the composition of the population of moths. How many are light color moths and how many are dark color moths? Early on, when the trees were light color, most of the moths were light color. Industrial Revolution, what happened? When pollution started, basically the only moths that you would find in the environment were the dark color. And notice the change, significant change. Far forward back 100 years to 1995, with the Clean Air Act and other pollution control devices in place, we go back very close to what we had in the past. So we are not doing anything to the moths directly, but their survival is different. And why do they survive? Because like here, this is a light color moth. If you look at the pictures, there are nice pictures in your textbook, you can see it better. Here I have a light color, someplace here there is a dark color. I don't see the dark one. But if I'm a bird that they eat them, you obviously are gonna eat the ones that stand out, that cannot camouflage. So the birds are the selecting factor, basically. The birds are the ones that are killing them based on their color. But this whole process of changing the color, the survival of the different colors was called the industrial melanism. So, pretty good example. Now you say, oh, it's just the gene for color, who cares? Well, remember that a lot of genes are linked. And when you start selecting for one gene, basically all the other ones that are linked and with low recombination rates, that starts changing too. So you might be selecting for one thing, but there is some baggage, basically, that comes with selecting for one particular gene that changes the overall genetic structure of a population. And what is evolution? Change over time. So again, selection factor here were the birds actually eating these moths. to finish up, just want to bring your attention to something that humans have been doing for about 10,000 years, since the advent of agriculture and when we started settling down. Here you have something that you might like or might not like. Eat broccoli, Brussels sprouts, cabbage, kale. That's a new craze this year. Kale, collard greens, all those. This is the same plant species. It's the wild mustard. And that has been selected, actually, artificial selection for plants that focus on different aspects. Some plants produce big leaves, yes, collard greens and kale. Some leaves are smooth, collard greens. Some leaves are ruffled, kale. Sometimes the leaf buds don't open up because there is a mutation and they stay closed. Hello, Brussels sprouts. When you eat broccoli or cauliflower, you are basically eating the flower buds. These are buds that are never gonna open, the cauliflower. The broccoli, actually, if you open every one of those little broccoli things, you're gonna see inside stamens and the different parts of the flower. So you're eating baby flowers there, baby reproductive organs of the plant. So artificial selection to different things. Starting with this. This is what the wild started like. And by selection, favoring the reproduction of different plants, because there is variation, we end up with these different types that we eat. For the record, this is what you are going to be planting and doing an artificial selection lab in class for the next 2 or 3 months. So this is your wild mustard. Artificial selection, we should end probably with the wolf. Again, wolf, we all know what we can get from crossing the wolf. And again, is the concept that if you decide which organisms get to reproduce, you can basically uncover all the variability that is hidden inside an organism. It's all because of the differential reproduction. It really matters who gets to reproduce. And that's the bottom line. We did the example of the peppered moth, and now I promise this is the last one. You know, Darwin, most of the ideas in Darwin, we started with Darwin, we have to finish with Darwin. Most of their ideas for Darwin came from the Galapagos, and in the Galapagos he studied the finches, little like sparrow-looking birds, but each island had a different type of species, and even within an island there were different species depending on the type of plants and the environment in the island, and they all have different beaks. And the different beaks were basically adaptations to the different types of food that grew in these islands. Now, where do those finches came? You are a set of young islands in the Pacific that just were formed by volcanic eruptions around 5 to 7 million years ago. The only way a bird is going to get there is flying from the mainland. So the closest relatives for the finches in the Galapagos are the finches in Ecuador, which is the closest country. Just for the record, so you think about these things. Things just don't pop up, they have to colonize places. Now, the finches. So people are still looking at these finches and still trying to monitor evolution and see how it's happening. And there is this couple, Mr. and Mrs. Grant, that had been working in the Galapagos for a long time, and they keep on measuring and looking at the shape of the beaks of the finches. And what they have discovered is, well, weather patterns change, of course, and in years where there is a drought, when there is less water, the seeds produced by the plants are thicker, thicker shells and slightly bigger. Thicker, because for protection, so they don't get destroyed. So in years that are droughts, less water, less rain, the seeds are thicker. Now if you're a bird, what do you need to have to be able to crack a seed that is thicker? Obviously, you need to have a stronger beak. If you have a wimpy beak, you break your beak, not the seed. So, they said, okay, if we go back and check the weather patterns and we match this with all this data that we have about big sizes, I bet you, and that was their hypothesis, the peaks will be probably bigger in those years where there was a drought that resulted in thicker seeds. So they picked all their data, they grabbed all the climate data that they had, and guess what they found? This is kind of the thickness of the beak. Notice that it shows waves. When they analyzed this and matched it with climate data about amount of precipitation, what did they find? Beautiful correlation. Years with thicker beaks match perfectly with years where they had a heavy drought. Why did they get thicker beaks? Only those with the thicker beaks or bigger beaks were able to survive cracking those hard seeds, so they are the ones that pass those genes to the next generation. Differential survival, only the fittest, new concept, are going to be able to reproduce and pass those good genes on to the next generation. Right? So I think we are done. I hope you get a big view of evolution and what we are going to be doing the next of this unit. And let's remember that we are going to start incorporating the concepts from previous units, like all the molecular data, so please keep that fresh in your mind. I hope this helps. Until next time.